Hi, everybody, and welcome. My name is Elchanan Bornstein. I'm an associate professor at the Department of Genome Science, and I'm the organizer of this lecture series this year. Before we start, I just want to apologize for having to cancel last week's talk. Cole Trapnell, our planned speaker, had a familial emergency and had to fly home. One of the problems with this seminar is that we send the announcement out there, and we don't really know how people get them. And so it was a bit hard to actually make sure that we got to everybody and make sure that everybody knows about the constellation. I think we managed to get to most of you. For those few that came in and only realized then that the seminar was canceled, again, my apologies. We're trying to run this as smoothly as we can, but sometimes things like that do happen. Having said that, it is my great pleasure to introduce our speakers for today, Dr. Aimee Dudley. Dr. Dudley is an associate investigator in the Pacific Northwest Diabetes Research Institute and an affiliated faculty also in the Department of Genome Sciences. I first want to give you a bit of info about Aimee's background. So she did her bachelor's degree at University of Massachusetts at Amherst, followed by a PhD in genetics at Harvard Medical School, and then a postdoctoral fellowship at George Church Laboratory, again in Harvard Medical School, before realizing that the Pacific Northwest is a much better place to live. So she moved here first as an assistant professor in the Institute of System Biology here in Seattle, where she was until about 2 years ago when she moved to the Pacific Northwest Diabetic Research Institute. Aimee's research is focusing on really one of the most fundamental questions in biology, which is often referred to as a genotype-phenotype problem, or in simpler words, can we try to understand and predict how the genetic content of an organism eventually translate to the morphology, behavior, and all the other phenotypes that we identified in organism. Aimee is not studying this directly in human, but rather in one of the most common model organisms, the baker's yeast. But I think what's really unique about Aimee's research is that while the vast majority of yeast geneticists are focusing on a small set of lab strains, Aimee is actually going and collecting wild strains of yeast and trying to identify the natural variation in those strains, and she will tell us a lot about this later on in her talk. Before inviting Aimee to the stage, I just want to share with you one story that I think is very relevant both to this talk and to this public lecture series in general. And this is a story that actually did not happen to me, but to one of my colleagues, Dr. Maitreya Dunham, another faculty here in the Department of Genome Science. And so Maitreya was traveling to a conference a couple of years ago outside of the U.S., and on her way back at the airport, she had to visit the immigration booth, and of course the immigration officer has always asked her, where are you coming from, and what have you done, and what are you working on? And Maitreya told him that she was a yeast geneticist, and to that, the immigration officer responded by, did you know that you can find yeast on chocolate and coffee? She was, of course, very surprised that an immigration officer knew this relatively little-known fact, until she realized that Aimee just passed through the same immigration booth a bit earlier in the day. I think what was really astonishing is that when she actually talked to Aimee later, she realized that Aimee did not pass there a few minutes before her, but actually much, much earlier in the day, several hours before she got to the airport. And if you can consider the number of people that the immigration officer talks to and see along a few hours, I think it is an astonishing fact that he still remembered this specific piece of information about the yeast. And to me, it's really a great testament both to the excitement that Aimee has in her research, but also to her passion to share this research with the public, which is really what this whole public lecture is about. And so with that, I hope you all help me to welcome Aimee to the stage and for a great talk.
Thanks, Elchanan, for that nice introduction. It's a pleasure to be here, and it's a pleasure to see so many people come out to hear about yeast on a nice sunny evening in Seattle. And so today I'd like to tell you a little bit, or maybe a lot, about my favorite organism. And my favorite organism is one that you're actually pretty familiar with if you've gone to the grocery store or you've made bread. And that is the organism Saccharomyces cerevisiae, which is baker's yeast. And so here you can see a packet of Fleischmann's baker's yeast. And if you've ever opened up one of these packets, you'll see that there are these little pellets inside. And those little pellets, we know thanks to a couple of undergraduates who are working in my lab, each contain about 3 million yeast cells. So each of these is not a cell, but in fact a whole bunch of 3 million cells that have been dried together. And we also know thanks to Trey and Marutz, that 77% of them are alive when you actually stain them under the microscope. And so not under our microscopes, but under very fancy microscopes, these are what actual baker's yeast look like. And so baker's yeast we know a lot about for one reason, because they're involved in the production of a whole variety of foodstuffs, both food and drink, everything from bread to wine to beer, sake, coffee, and chocolate, or as I like to call them, the 6 food groups. And so tonight, what we're going to talk about is using baker's yeast as a model, how microorganisms perform some of the important metabolic functions that can actually alter their environment. So microorganisms are micro, so they're very small, but they can actually have profound influence on their environment. Microorganisms also work together in communities of different microorganisms. So we're going to hear about different species working together to perform even more complex reactions or processes, and in a way that is reproducible across time and space. We'll then move to talk about how genetically distinct individuals, so Elhanan referred to this a little bit, how genetically distinct individuals from the same species, in this case yeast, actually have different traits that are influenced by the genes that they carry. And then finally, we'll spend most of our time talking about how scientists use the tools of genomics and population genetics to study a whole variety of different problems. And what I hope you'll remember throughout this talk is that we're going to talk about all of these topics in the context of something fun and light for a summer's evening, which is the production of food and beverages. But everything that I'm talking about is going to be relevant to microbes and microbial communities in the ocean, in the soil, and in our bodies. And in some cases, those organisms are exerting really helpful and beneficial properties. And in some cases, especially when the human body is immunocompromised, they can lead to sicknesses. But since I'm standing here in the city of Seattle and the University of Washington and the Department of Genome Sciences, I can't let you go away without telling you one tidbit about the history, the importance of Seattle for the yeast genetics community. So a yeast researcher named Herschel Roman was one of the founders of the field that I'm proud to be a part of, which is the field of yeast genetics. And Herschel Roman worked right here. I never had the opportunity to meet him. Also, if you came in on that side of the building, I hope you had the opportunity to see Dr. Lee Hartwell's Nobel Prize. Lee Hartwell won the Nobel Prize for work that he did here at the University of Washington and in what was then the Department of Genetics, but what is now this department, in the early 1970s. And Lee Hartwell, Paul Nurse, and Tim Hunt actually shared the Nobel Prize for the discovery of fundamental mechanisms controlling the cell cycle. And Lee Hartwell worked in the baker's yeast that we're going to talk about today. Paul Nurse worked in a different species of yeast called Schizosaccharomyces pombe. And the really important part, in addition to the historical aspect of this, is that this is one of the reasons why there are so many biological researchers in a variety of fields who actually study yeast, why it's one of the most popular model organisms. And that's because yeast is an excellent model for the study of processes that are shared between all the way up from microbes to humans. So many, if not all, of the processes that control the cell cycle, for example, how cells grow and divide in yeast, are conserved all the way up through humans. They function in the same way up through humans. And so by studying yeast in a much simpler system, we can understand problems that are important, for example, for cancer. In terms of my lab studies yeast for 2 very important reasons. The first reason is we're interested in understanding this genotype to phenotype problem. And that is the fact that we know that the vast majority of individuals within a species share the vast majority of their DNA sequence. Their DNA sequence is in common. However, there are, between any 2 individuals, a number of differences in their DNA sequences. And in some cases, those DNA sequences control traits like color or cuteness, or a variety of other traits, such as propensity for disease or response to a particular therapy. And if we could sequence these individuals, which we now can, thanks to the Human Genome Project and additional technologies pioneered by people like, for example, Jay Shinduri, who's in this department, there's the potential to actually try and relate genotype to phenotype. That is, to be able to predict how an individual might respond to a given therapy. And so this is actually the fundamental premise of human genetics, but unfortunately, we're not there yet. We can't do this really hard problem. And so what my lab and other labs like Maitreya Dunham's lab do is to use the simple organism Baker's yeast, where we can do experiments very rapidly, develop new technologies, to try and understand simple traits and even more complicated traits, which I'll tell you about today. In the process of understanding those biological processes, also develop new technologies, which can then be applied to, for example, human systems. The second reason that my lab studies Baker's yeast is that I've told you that Baker's yeast is pretty similar to humans, but it's even more similar to the other fungi, not surprisingly. And so some of these fungi are actually opportunistic pathogens that, if you have a compromised immune system, can be very dangerous for you. So Candida albicans is a species of yeast here, another species of yeast, and it's the fourth most common cause of hospital-acquired serious bloodstream infections. It's also the second most common cause of death in extreme low birth weight infants, despite antifungal treatments. There are other types of fungi. The molds are also a fungi, so most people have seen mold, right? And this mold, Aspergillus, and we'll hear about some Aspergillus later, is really common in the environment. It's all over the place. And unless you are immunocompromised, for example, if you've had an organ transplant or you're being treated with certain types of chemotherapy, it's not harmful to you. But for some of these patients, their mortality rates of 60%, or if you're unlucky enough to get infected with a drug-resistant strain, up to 90%. Unfortunately, antifungal therapy doctors have a really small arsenal of drugs. Some of those drugs are extremely toxic. And like bacteria, there is the emergence of resistance to many of these drugs. So in my opinion and many other people's opinion, we desperately need new antifungal drugs. And work in my lab is hopefully working to identify some good candidates. So let's get on to talking about food. And so the first topic is how microorganisms perform important metabolic functions that can alter their environment. And so here we turn to one of the earliest ways in which humans harnessed the power of microorganisms, and that is in making bread. So bread making began in the Stone Age. And people were making kind of pastes of grains and water, and they realized at some point that if they let them to sit over the course of several days, that they would start to become bubbly and inflated with gases, and that process could make the bread more palatable, and it could also make it, in some cases, more nutritious as well. And so bread making kind of took off, and by the late Stone Age, flat breads, like pita bread, for example, were extremely common. However, when most of us think of bread, we think of kind of a big loaf of bread. And that type of bread actually depended not just on the yeast, but on the development, the evolution of a certain type of wheat. And so that wheat became available, or had evolved, by about 8,000 BC. And the first archaeological evidence of leavened breads is about 4,000 BC. So how is yeast actually executing this process? And so one of the ways in which yeast helps in making bread is this fundamental aspect of its metabolism. So what yeast is really good at is taking a simple sugar, so one molecule, one unit of a simple sugar called glucose, and converting it to 2 units or molecules of ethanol, plus the gas carbon dioxide. Okay, so if you were making beer, ethanol would be the alcohol that's in the beer, and the carbon dioxide would be the bubbles in the beer. And when you're making bread, the bubbles make the holes in the bread, and they also leaven the bread. And then the alcohols and some of the other products that alcohol and other metabolites that are secreted by the yeast get converted into, those actually help together with your kneading the bread and the bubbles kind of expanding the bread, those actually help the gluten form. So they're forming the texture and the structure of the bread itself. So yeast is having a pretty profound impact on this kind of mush of water and wheat. But but microorganisms, when they work together, and even together in really complex environments, can have even more profound impacts on foodstuffs in this case. So as I said before, yeast is really good, baker's yeast is really good at taking simple sugars and converting them to ethanol. Okay, and that's one of the main features that people use it for, actually, knowingly or unknowingly. And so when you take grapes and crush them up and you mix them with yeast, either yeast that you've added or the yeast that are present in the vineyard, they will convert those sugars to alcohol and you get wine. However, yeast can't take the complex sugars. So starches are complex sugars. They're chains of these simple sugars. And yeast doesn't have the ability to break these complex sugars down into a more simple sugar that it can turn into alcohol. So if you'd like to take your rice and convert it into sake, you're kind of up a creek if you're just adding yeast, because you're just going to get rice plus yeast. However, what sake producers realized many hundreds of years ago is that if they take the yeast and mix it with a mold, a koji mold, which has this scientific name, and you put the two of them together, you have the yeast plus the mold, and rice plus the mold, and you get moldy rice. However, this moldy rice is actually the starter for making sake. And what the sake producers do is they mix the rice, which has the complex starch, the aspergillus mold, which is able to break that starch down into simple sugars, but not make ethanol, and then the yeast can then take that simple sugar and make ethanol from it. And actually, it's kind of interesting because in sake production, this ethanol production gets to extremely high concentrations. So sake yeast are a little different than, for example, wine yeast and some other yeast in that they're extremely ethanol tolerant. And so that's an example of another trait that you can start to think about being different between different strains or varieties of yeast. Okay, so that is a man-manipulated process. So the sake producers are consciously mixing the molds and the rice and mixing the yeast and the rice and putting them together and mixing them over many days. But let's talk about a process that's more than two organisms and that's a natural process that gets seeded naturally and then is carried forward. And this is the production of the fermentation that happens to make chocolate. So this is a cacao tree, and they're really interesting. You can see here, so these are the cacao pods that are going to produce the chocolate beans. And they actually grow off the bark of the tree, which is really interesting, unlike kind of the way most fruits, for example, grow off the branches. And they're really lovely. They have this lovely shape. They're about this big. And what a chocolate producer wants to get out of these pods are these beans. And these are about the size of an almond. But to get from this to this and then produce chocolate from it, what you first have to do is machete through a pretty hard skin. And then what you're left with when you open this is this mucilaginous pulp. It's this really slimy, white, gooey stuff. And in order to make good chocolate, the first thing that you need to do is remove this mucilaginous pulp from the actual bean that you want to get. And that's done often by so you scoop this goop out of the pods, and you heap it on the forest floor and cover it with banana leaves, and you leave it there to do this natural fermentation over the course of 8 to 10 days. And what magically happens in that 8 to 10 days is that there's a fermentation that happens, and it changes the chemistry of that fermentation, changes the color and the taste profiles of those chocolate beans. And so that fermentation is actually a rather complex fermentation that isn't exactly well understood, but it's pretty well understood. So we know that there are what I like to call sweeps or successions of different microbes. So in other words, what happens is instead there are going to be 5 different groups of types of organisms here. The yeast, different classes of bacteria, and then different kinds of molds. Okay? And those aren't going to all come out at the same time and start working. Okay? So initially the yeasts come in, and those yeasts can work on, for example, certain sugars which are present in the fermentation, and they're going to produce a lot of ethanol. And then at some point those yeasts are going to burn themselves out, and the lactic acid bacteria, very similar to the type of bacteria that make yogurt, for example, those are going to start becoming more dominant and taking over, and there'll be a lactic acid peak. And so all of these different chemicals and temperatures and modifications that these organisms are doing happen sequentially, and they happen reproducibly. So remember, there's nobody here adding yeast and adding bacteria. This is all a natural process, and there has been an evolution of these different organisms that leads to, miraculously, the fermentations that give cacao its special qualities. So if these fermentations don't happen correctly, if something goes wrong, the chocolate tastes terrible. Okay? All right, so this is an example of kind of a natural series of sweeps of organisms kind of becoming more dominant, doing their thing, and then burning themselves out, and another organism kind of taking over. And again, this is happening in chocolate, but it also happens in other ecological systems and biological systems. Okay. So now let's talk about, we've talked about multiple organisms, multiple different organisms. Now let's talk about properties that genetically distinct individuals from the same species can have. And so you're all, whether you know it or not, aware of this idea of genetic diversity because you've seen breeds of things, right? So you've seen, here's a nice colorful example that I like, different breeds of chickens, right? So farmers have bred different chickens to each other, and the results of all of that natural breeding, there's no genetic modification here, the results of those naturally breeding these organisms together for generations and generations has resulted in a wide array of genetic and phenotypic diversity. So these chicken strains or chicken breeds, they have different DNA sequences, and they have different traits that you can see here. And that's not just specific to chickens. It's also true for tomatoes and dogs, and I'm going to tell you even for strains, breeds or strains of yeast. And so to tell you about this, I want to first introduce you to a little bit of the history of what we're going to talk about, which is winemaking. So the relationship between humans and winemaking is ancient and tight, and that comes into play in terms of this breeding of yeast for making good wine. So the earliest archaeological evidence for fermented beverages is about 7,000 BC. And wine was so important that when King Tut's tomb was actually excavated, you could see that a number of these vessels were actually made for storing wine, so it had to be pretty important. Those of you who are familiar with Louis Pasteur, one of Pasteur's early studies and important studies was a study that he did of wine, and there he was he visualized yeast in wine and actually did some studies on the importance of oxygen in relationship to yeast and fermentation. So kind of now to these wine strains. So winemaking was developed and perfected very early in human history. And so then humans started migrating. So winemaking develops, and then humans are trading and they're migrating to different places. And as they're doing that, they are taking with them not only the grapes that are involved in winemaking, but also the vessels for storing and producing the wine, as well as the remnants of previous fermentations. And the results of doing that are sometimes you make a batch of wine, and it's a great batch of wine. And early people realized kind of early on that if they then seeded a new batch of wine with the results of a good quality, the remnants of a good quality batch of wine earlier, that they would get a much better product. And so humans were taking not only just the grapes, but also all of the yeast around with them throughout the world. And the result of this is that actually when you sample wine strains, so if you sample strains from a vineyard in California, or in Argentina, or in South Africa, or Australia, or France, the wine strains will be extremely similar to each other. And that is true if you sample off the grape from the wine production itself, or even off of the soil that's resident in the vineyard. And that is because this yeast is so pervasive. And one of the reasons why this yeast is so pervasive and so similar has to do with the activity of humans kind of bringing this around and producing so much wine. Okay, and so, but you can find other strains of wine. Sorry, not other strains of wine. You can find other strains of yeast. So you can find yeast, for example, if you get away from a vineyard and you go to underneath an oak tree in North America, you can actually find native North American yeasts that are not the wine strains. So does it really matter? Was it really that important for early and current humans to actually work with these specialized strains of yeast? And so what one of my collaborators, Justin Fay, did was a really fun experiment, I think. And so what Justin wanted to do was to actually test that in the lab. So what he did was to order up a whole lot of Chardonnay grape juice that they sterilized. So there's no bacteria and no other yeast in this grape juice. And then they proceeded to make wine with either some wine strains that they had, some sake strains that they had, or some wild strains that they had from oak. And then they wanted to evaluate this wine to see how well it tasted. But, you know, Justin's not really an expert, so even though I'm sure he likes wine. And so what they did was to actually hire 6 experts who are trained in actually tasting and evaluating the aroma and flavor profiles of wine. And so they professionally analyzed these, the wines that were produced with these different strains. And so they analyzed, so wine tasters apparently analyze for some favorable properties like floral scent and notes of citrus. But they also analyze for some, I would imagine, unfavorable qualities like cabbage and wet dog. And so that is the first time I've ever said wet dog in a professional seminar. So this is a big moment for me. And so the result of that was really interesting. There were 2 important results. The first important result is that strains that were genetically similar to each other, for example, the wine strains, made wine with profiles that were similar to each other. Okay? So something about the genetics of those individuals, the individual yeast strains, are conferring similar properties to the wine. The second important point is that the wine strains actually make pretty decent wine. So you have these axes are the longer this line here, the more citrus you have, and the longer this line here, the more floral character you have. And those are for the wine strains. However, when you worked with Justin's oak strains, you got quite a bit of wet dog, you got quite a bit of mushroom, and a decent amount of cabbage. And so probably not my favorite wine. And so that speaks to the fact that there probably is a pretty big selection for selecting for certain strains of wine that human activity has brought about and transported across the globe. Okay, so I'm telling you that these strains are very similar across the globe, but I haven't told you how I know that. Okay, so let's talk about some of the tools of genomics and population genetics that scientists use to study these types of problems and many other types of problems as well. Okay, so we talked about genetic diversity before. Okay, so let's return to our chicken breeds. So if I take these 3 chickens and you look at them, they look pretty different from each other. They've got different feather colors, they have different body shapes. One might make more meat than another, one might lay more eggs than another. But if the trait that I'm really interested in understanding is, for example, egg color, you can see here that there's a whole variety of egg color. But my 3 chickens that I have access to that I'm going to study only have this really narrow range of this kind of light brown color in terms of the eggs that they lay. And so if it's really important for me to understand how you make a green egg or how you make a white egg, and of course, you know, the color of eggs may not be the most scientifically important problem, but you can see the principle in that, you know, you might want to make, for example, a yeast strain that makes really high levels of biofuel. So if I only have a limited number of strains and those strains don't actually undergo the process that I care about, then I'm kind of sunk. And so our lab ran into this problem when we first started the lab. And the problem is that the vast majority of yeast researchers, including myself up to that point, worked primarily with a well-characterized and valuable set of strains that had been propagated in the lab. And these have really good properties. They're really important for certain types of research. But my lab was interested in understanding certain types of problems. For example, you can see here that these wild strains, some of these wild strains, right? So the first thing that you can see is that they're much more diverse. They have different colors in some case, and in many cases they form these really weird shapes, right? And those weird shapes are also formed by some of those pathogenic fungi that I was talking about. And these pathogenic fungi, what they're doing is they're forming something called biofilms. And biofilms are the plaque on your teeth. Biofilms are also the way in which many pathogenic organisms, bacteria and fungi, can stick to medical devices. And in some cases, the ability to form these biofilms is correlated with resistance to drugs, right? So being able to stick to a medical device and being more drug resistant sounds like a bad thing. And so we wanted to study a lot of these processes and develop new techniques for doing so. But if we were working with the lab strains don't do this. And so we needed to isolate wild strains. Oh, just to give you just one little tidbit of the technologies that my lab has developed to study some of these biofilms. Here is a movie that was generated in my lab using an automated imaging system that Adrian Scott in my lab developed. And what this does is it actually is a camera system. And it looks like what we're doing is just sitting on a set of strains and taking a time-lapse image. But in fact, what this camera system is doing is zipping around the series of plates and taking images of thousands of these every 10 minutes or so. And what this gives us is high resolution to be able to study the specifics of how a single-celled organism organizes itself to form specific structures such that genetically identical individuals, so these are all the same strain, they're essentially identical twins, and you can see that now a single-celled organism is organizing in order to form a shape that's really so similar that you could almost overlay them in Photoshop. And then a different strain, this one over here, its identical twins also look similar to each other, but very different from the strain that it's genetically distant from. So that's a way in which my lab is applying technologies that we're developing to understand the formation of biofilms. But back to our problem. So the problem is that we have this collection of lab strains. We know that the wild strains are going to be valuable. We want to get more of them. So what Kathy Ludlow in my lab did was she knew that we could take environmental samples and that we could grow them in media, in nutrients that yeast really like to grow in, and we would get mostly yeast. But we would get a lot of different kinds of yeast, and we had to weed through them and identify the yeasts that were actually the ones we wanted, which are Saccharomyces cerevisiae, baker's yeast. And so sometimes there's not any baker's yeast there, and sometimes it's not there at very high levels, and it'd be really nice to be able to say, that's the one that we want. And so Kathy realized that actually clinical microbiologists have lots of tools for doing this, and one of the tools that they use are plates that actually change color depending on what the species of yeast is. So different yeasts have different enzymes, they have different metabolic properties, and so the baker's yeast that we study turns this little purple color right here, and one of the dangerous types of yeast, Candida albicans, turns this nice light blue-green color on these specific plates. So great. Kathy now had a system in hand that would allow us to quickly and easily go from an environmental sample to the specific kind of yeast that we wanted. And this was so quick and easy that a high school student could do this. And so we were really excited about that potential too. So now the problem is, okay, where are we going to find all these wild yeast, right? So if you wanted to go and find a new type variety of squirrel that you had never seen before, going and looking in your backyard would probably not be the best place to do that. Going and looking at the park down the street would probably not going to be the best place to do that, because you just find the same type of squirrels that you see around you. What you want to go and do is look in places where nobody has ever looked for squirrels before, or not very hard. And so when we looked at where the strains, the few wild strains that people had identified had been isolated, they were actually, other than the wine strains, which we know are going to come from vineyards all over the world, we're not going to look there. other than that, they had actually come from a really small region of geography. So, for example, almost none of the southern hemisphere had been sampled. And so we tried setting up collaborations. We tried working with the USDA to let us bring in soil samples and plant samples, and they don't like that. And so we realized this just wasn't going to work. So Kathy was busy planning her worldwide trip across the southern hemisphere. And unfortunately for her, she had an epiphany. And that epiphany was that there are lots of foodstuffs, actually, that are, other than wine, that are the process of fermentations. And some of those foodstuffs are grown in really interesting and exotic places that we would like to sample. So one of those is actually cacao, which we were just talking about. And one which I'm going to focus on just in the interest of time is coffee. And both of those foodstuffs are fermented on site and then completely legally and with all the blessings of the USDA imported into the United States. So we were good. And fortunately for us, we're living in Seattle, so there was a roast your own coffee bean fad going on at the time. And it's also relatively easy to get unroasted cacao beans. And so Kathy did that. And what she was hoping that would allow her to do is to be able to sample places where, for example, coffee is grown. So coffee is actually native to Ethiopia, and wild examples of the plant still exist there. But in about the 6th century, it was brought to Yemen. And then Arab traders disseminated it throughout the Arab world in the Middle East and North Africa. And by the 17th century, it had made its way to the salons of Europe. And then Europeans were hooked once they realized how to roast it and mix it with just the right amount of sugar and milk. And cultivation rapidly spread across the entire coffee belt, which is the region in which coffee is able to grow, and that is between the Tropic of Capricorn and the Tropic of Cancer. And so these are all places where we really have very poor coverage of our yeast strains. So Kathy wanted to know whether or not we could actually isolate live yeast from imported foodstuffs. So can we take yeast that have been fermented can we isolate yeast from green coffee beans that have been fermented all across this coffee belt, bring them into Seattle, and get live yeast out of them? When we started this project, it wasn't clear if that would be the case, but of course I'm telling you this because it was. And in doing so, this allowed us to very quickly and very easily isolate lots and lots of yeast cells, different yeast strains, from lots of places in Central and South America, in Africa, Indonesia, and also in lots of islands. And geneticists are kind of intrigued, and evolutionary biologists are kind of intrigued with islands. And so this was really great. And so we've got a whole collection of yeast, wild yeast, and we're working with them. But one of the things that we were wondering, especially because we were working with Justin Fay, who's a yeast population geneticist, and one of the things Justin thinks about as a yeast population geneticist is where did these yeast come from, right? So they obviously came from the place where we isolated them, but where did they originally come from? How did they get to Papua New Guinea? How did they get to Yemen? And so there are two kind of models for this. One is the wine strain model, and that is the thing I've already told you about, which is as humans brought around the plants for making wine, they also brought with them the yeasts that were really good at making wine. So that's one model, is that maybe as people are transporting chocolate plants and coffee plants, they're also bringing along the yeasts that are really good, and maybe other microorganisms that are really good at making coffee and chocolate. The other is kind of what I'm calling the oak strain model, which is in North America, you find oak strains. They're out in the wild. They're native to North America, and there's nothing that humans have done that we know about to really bring them there or to transport them to other places. They're the native yeasts that actually live in the place that you're sampling. Okay, and so we're talking about, in order to kind of explain to you how we figure this out, I need to take a quick detour. And that detour is actually deals with how we deal with geographic signatures, okay? So there are I'm going to tell you about geographic signatures in DNA sequences, but there are geographic signatures in all kinds of things. Okay? So I'm from Boston, and I was born in Boston. I grew up in Boston. I lived in Boston for a long time. And when I am driving, I come to something like this. I call it a rotary. And according to a dialect study that a group of Harvard researchers have done, the answer to that specific question pretty much pins me somewhere in New England and probably in Massachusetts. Similarly, if I'm at my kid's school and I want to get a drink of water, I go to find a bubbler. And a bubbler puts me either somewhere in Wisconsin or somewhere here in Massachusetts or Rhode Island. And so if you take together not just the fact that I call something a rotary or a bubbler, but you put them together, you can actually get a more precise signature of where it is that I'm from. And that's because those word choices are particularly informative. If, however, you were to ask everybody in this room, what do you call this animal? Everybody would say either a cat or a kitten. But whether you said cat or kitten doesn't determine anything about where you're from. And similarly here, we would say puppy or dog, and everybody would say puppy or dog. These are what we would call uninformative choices. And so there are about 1 million words in the English language. And so and there are about 3 billion bases or letters that are in the human genome. And again, the vast majority of them are the same. We use the same words to describe most things. And the vast majority of our DNA sequence is the same. But there are some differences, and sometimes these differences are informative, and sometimes they're not informative for the thing that we happen to be studying. And so sometimes your dialect is informative about where you came from. And sometimes your DNA sequence is informative about where you came from, in that it's talking about your ancestry. So there was a really nice study, which I'll tell you about in a minute, where they actually looked at the signatures of DNA sequences and how they might relate to the origin of certain people in Europe. And so just a reminder, the genome sequences of every human in this room is well over 99% identical. The vast majority of our sequence is completely identical. But the genome sequences of these two kids right here is much more related than the rest of us. And that's because they're related to each other. Okay? So related individuals are much more similar to each other than their genomes are much more similar. And so before we all had modern transportation and we all went away to college and we all, did all of these things that meant that we met other people and moved away for our jobs, for example, people tended to stay where they were born for generations and generations. And the result of that is that people from common locations became more and more related. Right? So, maybe you have these different families that live in this little village, and now the yellow family decides, I don't want to live in this little valley anymore. I really want to go raise mountain goats, and I'm going to go up over on this mountaintop. So most of them kind of disappear, and so the special changes in their DNA sequences kind of disappear from the population. And over many, 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 many generations, that can lead to a geographic signature of where you're from. And one study that I really like took individuals, and they had to be specific individuals. You can't do this with just anybody. They were individuals whose grandparents had been born in the same little village in some European country, and all 4 of their grandparents had actually come from this same little village. And so, and that meant that their family had lived there for a very long time, and everybody from their gene pool, if you will, had come from that village. And when they did this, the really interesting thing was, so they sequenced these portion of these people's genomes, and then they just grouped them together by how similar their DNA sequence was. And the thing I love about this is that this, so 2 points here are close together if they're related to each other, and they're far away from points whose sequences they're not. They have more differences between, right? So these 2 individuals have more in common, and these 2 individuals have more informative sequences that are different. And when you do that, you find that this groups together Spain and France and Germany and Great Britain and Italy, and it looks like the map of Europe. And in some cases, again, only with special individuals who have been kind of selected in this way, you can actually, and in certain places, you can actually identify really fine, this on a fine scale. So distances of only 100 kilometers, right? So they were actually, in this particular case, able to take, and just by their DNA sequences, group together the French-speaking Swiss, the German-speaking Swiss, and the Italian-speaking Swiss. And that's because those groups tended to stay together and marry with each other and live in the same places for a long time. But these are examples of geographic signatures, right? And how your DNA sequence can have, in some cases, a signature of where you're from. And that's all well and good for humans, but what about the really important thing, which are our coffee and cacao yeast strains? So we took and applied those same types of methods. So we sequenced portions of the genome of our coffee and chocolate strains. And so if the wine model is true, then all the coffee strains will look similar to each other, and there won't be any geographic way to group them together. But if the other model is true, then they'll group together by geography, regardless, maybe not regardless of whether they're chocolate or coffee, but there'll be some geographic correlations there. And so the answer to that is, ta-da! Actually, the story with coffee and chocolate is actually very different than it is from wine. So coffee and chocolate, the fermentations that happen for those are actually not common to and consistent with a model in which the yeast strains got carried along with the plants. But in fact, those fermentations are happening with wild yeasts that are actually endemic to the place in which those plants have been brought. Okay. So one last experiment that we did, which was kind of fun. So that correlation with geography was so strong that we had one or two examples where we had a strain that we had isolated from coffee that said, this coffee is from Yemen, but it was clustering with Colombia. We subsequently found out that Yemen coffee is actually quite highly prized. And so it looked like there was either an innocent mistake or a not-so-innocent mistake in terms of labeling those coffee beans as coming from a place where they actually didn't come from. And back when I was a kid, there was a series of commercials called the Folgers Challenge. And so it was a blind test where people were given two types of coffee, one really cheap coffee, Folgers, and one really expensive coffee in some fancy restaurant. And the participant had to tell which one was the expensive coffee and which one was the cheap coffee. And so we thought that this correlation with geography was so strong that we would actually be able to tell you, based on the sequence of the yeast strains we had isolated from the coffee, where it came from. So what we did was we asked a coffee supplier to give us 3 bags of green unroasted coffee beans, and to not tell us where they were from or anything about them, but to write down their country of origin of the beans in a top-secret envelope. This is not the envelope. But just to write down where they were from. And we wouldn't open the envelope until we had isolated yeast strains, sequenced them, and put them through our analysis pipeline. And so it turned out that there are some coffee strains, some coffee samples that we actually can't isolate yeast from. So because we didn't tell this supplier anything about our experiment, he actually didn't know that we're actually unable to isolate yeast from coffee that's been processed in a certain way. So that's likely what these are. However, from the 4 samples that we were able to isolate multiple yeast strains from, we predicted that 2 of the strains would actually come from Nicaragua or Honduras, and they're very close to each other, so we can't distinguish between those 2 countries, Peru or Rwanda. And in fact, 100% of the time for this small set of samples, we were able to accurately predict, based on the sequences of the yeast that were associated with those coffee beans, where they had actually come from. So just a final food for thought, if you will. I hope today that you've gotten an introduction, at least, to why studying microorganisms like yeast is interesting and important, as my old boss used to like to say, and still does like to say. An appreciation for the complexity of microbial communities and the processes that they can carry out. an understanding for the importance of studying genetic diversity and the need to develop new technologies and new methods to be able to do genetics better, as we like to say. And if you got nothing else out of this talk, I hope you got an interest in learning more about genetics, genomics, and how they're being applied by scientists like the scientists in my lab to solve hard and interesting problems. And one final note, the most important note, is to thank the people in my lab at the Pacific Northwest Diabetes Research Institute who did this work, our collaborator at Washington University in St. Louis, Justin Fay, the people at the government, your tax dollars, who fund, in a lot of cases, who fund this research. And for those of you who are over 21 and who enjoy good wine and good food, I'd like to put in a small plug for the PNDRI's annual Evening of Wine, which is a fundraiser that supports research like the work in my lab. And I'd like to thank you for coming out on a nice summer day.
So we have time for a few questions, and just to remind you, like every week, Aimee would kindly agree to stay after the talk, and so if you do not have time to ask now, you can ask it after that. Some coffee, probably not as good as you want to describe, and cookies.
Yes?
Can you talk about how these strains have to do with cancer?
Yeah. So you can actually use yeast as a cellular model, what we call a model system for studying cancer. And you can do that in a lot of different ways. So, for example, some of the processes that are important for cancer, so one of them, for example, the process that Lee Hartwell and others were studying and still do study, actually, is the progression through the cell cycle. So there are certain proteins that, when they malfunction, will lead to the progression of cancer. And those proteins exist in yeast, and they also exist in humans. They exist in yeast and mice and cats and dogs and humans. And they have pretty much the same function in yeast as they do in humans. And so one of the ways in which you can use yeast to understand that is that in yeast, I can do an experiment every day. And yeast don't have any feelings, sadly. Well, not sadly. It's actually good that they don't have any feelings. You can perturb yeast, and you can do all sorts of things that would be impossible or unethical to actually do in humans. And so in this way, you can actually quickly understand what those genes that are involved in cancer, right? So we might, for example, find a linkage between having mutations in a certain gene and a propensity for cancer, but we don't know what that gene does. We can either study it in yeast as the yeast gene, or in some cases, you may have heard in the news recently, one of my yeast colleagues, Ed Marcotte, has actually put many of the human genes, in fact, a really large collection of the human genes, actually into yeast and used yeast to do what we call assays, tests of whether or not certain mutations will actually affect the activity of those proteins.
So if I take your example of wine as a model, where you said that if I have a good year and I get good wine, and I take the yeast and put it into next year's wine, I'm going to get a good wine. So can you do the same thing with coffee? Can I take let's say, an expensive high-end coffee, Jamaican Blue Mountain, go and isolate yeast, use that to ferment a low-end coffee and get Jamaican Blue Mountain coffee out of it?
Yeah, that's a really good question. So in chocolate, so the coffee industry is a little bit behind the chocolate industry. But in chocolate, there is a lot of interest from some very high-end chocolate producers in doing exactly that. So taking some of the microbes that are responsible for, or at least resident in, high-end chocolate and using them to seed fermentations. And, you know, it works to some extent. So there's something called terroir. Right, so it's a very fancy word that foodies like to use. But terroir involves everything about the place in which the the coffee or the chocolate, for example, is grown. And that has to do with the microorganisms that are present in that environment. It also has to do with the growing conditions, how hot, how wet, and other aspects of the soil and the environment there. And so one of the things I really liked about Justin Fay's experiment is that it's not clear, right, because that terroir is such a complex system and it changes from year to year. So by going into the lab and testing how much of an effect was there of just the yeast, of just this one organism, where in coffee and chocolate we know that there are many, many other organisms.
So is it clear why the wine model doesn't hold? Why when they moved coffee from Ethiopia to Mexico,
Yeah.
for example, why didn't the yeast come with it?
Yeah, I think because, number one, they weren't moving the remnants of the fermentation or the vessels that are involved in making the fermentation. So when you make wine, right, one of the reasons why you can isolate those wine yeast just off the floor of, not the floor of the vineyard, but the soil in the vineyard, is that there is so much yeast produced. And there is so much remnant of that fermentation.
But I must have translated, I must have transferred green beans, and that's what you're using to isolate your yeast.
When I'm transferring the coffee plants to start my plantation, I'm not bringing a huge vat of the fermenting coffee cherries with me necessarily. So there are yeast getting moved around. So, for example, we can find signatures of, for example, those North American oak strains or some of those Asian strains. We can find those mixed, for example, with the wine strains. So part of their sequence is like wine, part of their sequence is like oak, and those are called admixtures. Okay, and so we see examples of those as well. Yes. Mm-hmm. Mm-hmm. Yeah, so in some cases, for example, with bacteria, there's kind of very good what I would consider direct evidence that the ability to form a biofilm is actually related to the actual pathogenesis. In the case of, for example, Candida albicans, which is a yeast, and it's the most common yeast fungal pathogen, the evidence is, I would say, correlative. That is that we have sick patients, and we isolate a lot of Candida albicans from lots of individuals, and we see that in general, the ones that can form biofilms tend to be more drug resistant. And so there's a connection there, and it's not terribly well understood. So this falls into the category of places where active research is being conducted and is needed. But so I can't tell you the exact answer, but what I think is that actually we know, my lab is studying many of the pathways, and actually Maitreya Dunham, who's another researcher here in the Department of Genome Sciences, both of our labs are studying many of the pathways that are related to biofilm formation, the ability to adhere to medical devices, and also to kind of invade and eat through, in some cases, medical devices and other substances, surfaces. And those processes, the cellular pathways that control those properties are actually some of the same pathways that are signaling to drug resistance. And so that's why I think that there is a correlation, but not a perfect one-to-one relationship. But this is a case where only through studying these in real detail can we start to get at some of those answers. Thank you very much.